Okay, it's 9:30. Good morning, everyone. And this is the first of the mini-series on immunology. Immunology is a very complex subject, so for all those who aren't here, so there is no chance, I think, one can learn this in 24 hours. So I recommend starting these lectures as early as possible, ideally being in the lectures. So it's also has some sort of network, and it's not so linear, so you can basically ignore most of the stuff on bacteriology and still be a champion in virology, but such a thing is not possible with immunology, so as you will see. It's also sometimes a little frustrating if this is the first comprehensive view on the subject, because sometimes you just have to think and take things for granted, which will become clearer in a subsequent lecture. I will do my very best, and you can stop me at any time and ask questions if you like, and let's see how we go. So the first lecture, it's going to be an introduction, and it's going to focus on what arms of the immune response are fast, which are slow, what is general, and what is specific. So let me get the pointer up. Here we go. This is on. Lights are down. Okay, and I can only highly recommend our textbook. Even if I wish that they would publish a new edition, I think it's one of the textbooks that has the most student-friendly figures. So that's why I really like to stick with it. Okay, so this figure is not in the textbook, that's mine, and it's a bit of an overview about the innate immunity and the adaptive immunity. And you could look at it in a different way as the first, second, and third line of defense. So that also brings things in order that are, things that are faced by potential pathogens immediately, after a quick response, and after a slow response. And if we start with the first line of defense of the innate immunity, so these are mostly passive barriers that are there all the time, and they may have other functions, but they also have benefits in warding off pathogens. So for example, the skin protects the interior of our body because everyone who wants to get in needs to go through the skin, which normally is not easy or impossible and requires cuts, abrasions, penetration by a blood-sucking mosquito, something like this. So the skin is fairly active. There are also enzymes present on, near, or in these barriers, and I will discuss one, the lysozyme, because it allows me to hark back on one of my favorite subjects. And funny enough, there is a normal body flora. We possibly encourage, not just tolerate, things that live in and on us, because they keep more nasty things at bay. So they basically occupy the niches. And this is one reason why antibiotics, for example, should not be prescribed in an ignorant way and willy-nilly and all over the place, because apart from the potential risk of creating resistance in the environment, they also kill parts of the normal body flora, which then leaves the patient more susceptible after the treatment. Okay, and sometimes we restrict the presence of things the pathogens need, and for bacteria this is very famously iron. Okay, come to the enzyme, lysozyme. That's an antibacterial enzyme. Here is a depiction of the enzyme. What is depicted in yellow is not part of the enzyme, but part of the substrate. And this enzyme serves as a non-specific innate opsonin by binding to the bacterial surface, reducing the negative charge and facilitating phagocytosis of the bacterium before opsonins from the acquired immune system arrive at the scene. In other words, lysozyme makes it easier for phagocytic white blood cells to engulf bacteria, and the importance of this we will come to later in this lecture. It also, because it's an enzyme and just not only sticks and flags the bacterium must be in foreign, it can also attack peptidoglycan. And you know all by heart, hopefully, that peptidoglycan is an important component of the bacterial cell wall. And if this enzyme destroys peptidoglycan, it destroys the bacterial cell wall. And what it does, it cleaves between NAM and NAG sugars right here. And if it does so often enough, then the bacterial cell wall will become apart, and then the bacteria will become apart. And you know already that this is a gram-positive bacterium and the cell wall is exposed. So there is no protective outer membrane on top of the cell wall. So this is sensitive to the enzyme, whereas gram-negative bacteria are more resistant due to the outer membrane. So I show this again, reinforce again, that this is an important subject. Okay, now we come to the second line of defense. Now it gets a little more complicated. So that is mediated, for example, by cells that are always present. And I name a few, macrophages and natural killer cells, and I will discuss them in separate slides later. There are also cytokines. Cytokines are proteins that have messenger functions. There is the complement-mediated arm of the innate immune defense, but I decided to skip this altogether because we do not have time to go through this. So if there will ever be a proper immunology lecture series, then I will hark on the complements, but for now you can consider yourself happy because it's a very learning intensive subject, and we will not touch on this in detail. Okay, so here the second line of defense requires pattern recognition molecules. So the immune system has to detect that something isn't quite right. And then there is a response time. This is measured in hours. So this one is up within a day. Okay, so now let's look a little bit on these pattern recognition receptors. It's a bit of a German story, and you can tell because many words in this field are German. For example, one here, this is an excellent one, toll. Toll is Old German for amazing or mad. You would probably call a drunken Viking warrior who is just bloodthirsty and not reasonable at all, like a berserker, and an adjective to characterize this further would be mad. And you think, what has this to do with immunology? So there was someone, do we have an image here? Christiane Lüstlein-Vollhardt, who got the Nobel Prize for mapping genes for development and other purposes in Drosophila. And they had one, they thought, you know, this looks amazing or just crazy wild, and they named the gene that's responsible for the phenotype toll. And you will see they got much more out there in other phenotype namings. And this one initially was discovered as important in establishing embryogenesis, in particular the dorsoventral axis. But later it turned out that it does so by being part of a cellular signaling process. And you do not need to learn this. Here are very weird names if you speak the language. But this was the original story, and then no one really cared because they thought, oh yeah, this is something, you know, that drives the evolution of fruit flies. Is that of any importance to us? But then we had a Cell paper a paper in the journal Cell, which is possibly as high as it gets for the subject, that the gene also controls immune responses. In this case, it controls immune responses to fungal infections. And here we see a fruit fly that is not wearing fur. This is a fungus grown on the fruit fly, and it grows because the fruit fly can no longer express the toll gene. So then, a year later, it turns out that we, humans, have lots of those. So now they are called Toll-like receptors, and as you can see, we have kind of 10 or 11. And then research really kicked off, and it was recognized that these are pathogens, that the Toll-like receptors recognize pathogen-associated molecular patterns, or PAMs. So they do not recognize a particular protein on a particular pathogen species. They rather recognize classes or groups or certain patterns. So it's somewhat unspecific. So, and you see that the different Toll-like receptors, Toll-like receptor 1, 2, 3, 4, recognize different things. For example, Toll-like receptor 4 recognizes double-stranded RNA, weird thing. And double-stranded RNA of certain types is prevalent at abnormal levels in virus-infected cells. So this one, therefore, detects virus infections. Toll-like receptor 4 detects LPS, and that is present in abundant quantity on gram-negative bacteria. So it doesn't recognize particular gram-negative bacteria, it recognizes a class of bacteria. It also recognizes a particular virus. Here, this recognizes flagellin, so bacteria that do have flagella. And that kind of gives this arm of the innate immune response some direction. It's not very specific, but this arm of the immune response can distinguish whether you are threatened by certain bacteria, certain viruses, and so on. So cells also contribute to this arm of the immune system, and I would like to show you this slide on hematopoiesis, and this is to show that all the immune cells originate in the bone marrow. So we have like hematopoietic stem cells that reside in certain bones, highlighted here, and they kind of differentiate into all kinds of cells. And these are the final stages of the differentiation. So we have mast cells, basophils, eosinophils, neutrophils, monocytes, macrophages, and also the erythrocytes. And I will give you here an idea of how many of those you can find in blood, in human blood. So here, for example, we have the neutrophils, and there is a certain amount. And that means out of the white blood cells, so roughly every cell that isn't a red blood cell, they have usually more than half. And the second biggest group is lymphocytes, which are made of T cells and B cells, but just by looking at them, you cannot really see whether it's a T cell or B cell. And they make the vast majority of the white blood cells. It's actually quite important for diagnostic to determine whether a patient has more neutrophils than usual or less neutrophils than usual, and whether particular sites like eosinophils are up, because that gives you an idea about the nature of the infection. So again, because this kind of immune response is somewhat directed, so you can tell. Okay, here I would like to show a little bit on these cells here. And the neutrophils, for example, are the first immune cells to arrive at the site of a bacterial infection. So here we have like a distinct example. So we have a tissue damage. This could be a rusty nail or a knife. And so we penetrate the skin. So this is the opportunity for bacteria, depicted here in green, to penetrate and to get into the tissue below. And they sometimes like it there because there's lots to eat. It's warm and it's protected, and there are most likely not any other bacteria. So they multiply, and this creates a problem. So the tissues underneath become leaky. So, and they are leaky because they have to allow immune cells from the blood to penetrate the tissue, so like these ones do, but that also leads to the leaking of fluid, and that's responsible for you experiencing this swelling pretty quickly after a cut or an abrasion. And then the immune cells start to release substances that make it uncomfortable for the bacteria. They also start to eat the bacteria, A, to get rid of them, and B, to process them and to produce bits and pieces on their surface to direct more specific immune responses later. So, and this slide is about the eosinophils. And here we have a schematic view and a real-life microscopic photo of an eosinophil, and the eosinophils are rather large. You see this in relation to the surrounding red blood cells. And they are relatively complex because they have a lot of granules. If you remember the scattering, a forward scatter and the sideward scatter, so these ones would be found on the top right of that plot because they are very complex and they're fairly large. And the job of the eosinophils, or one job of these cells, is to attack parasites. And here we have a parasite that is surrounded by eosinophils, and they're trying to load off their granules and to create a microenvironment that hopefully will kill this parasite. So, and now we come to another bit which I think is very important, and these are the natural killer cells. Quite a name. So here we have an example of a natural killer cell in a blood smear, and here we have a time course after an infection that kind of shows us what happens when. And the infection is an infection by a virus, and here we have the virus titer. So in yellow is after the infection the virus grows, and then the growth slows down, and eventually the virus titer is reduced, and in an ideal world, the virus is eliminated. And then you have the status quo ante, before infection. And here you see in green, blue, and red different immune responses. So in green, we have the production of these cytokines, these messenger molecules, and very important for fighting viruses is the early production of lots of interferon, IFN. And then here we have the natural killer cell-mediated killing of infected cells. That starts very, very quickly because the natural killer cells are always there. They don't need to amplify. They don't need to increase in numbers. They are there just waiting for viruses to come along. And then lastly, in red, we see the T-cell-mediated killing of infected cells. And we see here that nothing really is happening in the first couple of days. And it really goes on possibly after a week. So there we have the maximum. So this one takes time. But this one correlates with the elimination of the virus from the body. So you may say this is the most powerful immune response. So this is the adaptive immune response. Here we actually have to generate the right, to select the right T cell and then to amplify it and then to let it off the leash to fight the viruses, or rather the virus-infected cells. However, without these responses, the patient may be dead before the patient was able to mount an effective T cell response. So all these responses are important, even if only the T cell mediated response eventually gets rid of the virus infection. Okay, now we come to monocytes, macrophages, and dendritic cells. So you see they are somewhat related, and they have important tasks in antigen presentation and in directing the adaptive immune response that, in terms of virus infections, heavily relies on T cells that kill virus-infected cells. And I have like a whole lecture after this one on the subject. So they are sometimes called professional antigen-presenting cells, or APCs, and there are many cells that are professional antigen-presenting cells. The most known ones, or the most important ones, if you like, are monocytes, macrophages, and dendritic cells. So here we have a schematic representation. you see that this is a funny looking cell because it has all these little arms that they call pseudopodia. So they increase dramatically the reach and the surface area of these cells. So, and for these cells, it's quite important that they have a large surface area because they have to make contact with things that are foreign, and the bigger the surface area, the bigger the chains. And later on, they have to make contact with other cells of the immune system and interact with them, which also needs a larger surface area. So apart from these major names for these major groups, there are other cells that also belong to professional antigen-presenting cells, and they are, specialized cells in certain organs. Okay, so now a quick overview of the phagocytosis of pathogens and the subsequent antigen presentation. So here we have a schematic view of one of these professional antigen-presenting cells. We see the pseudopodia here. We see that this is a cross-section. The brown bit in the middle is the nucleus. For what we are discussing now is not really important. So, and what is colored here in lighter is the cytoplasm, and here's the action. So what we see here in orange-red is one bacterium. I don't know what else is bacteria. And that is engulfed and eaten. So it's phagocytosed. That's the proper technical term. And then it ends up in a phagosome. And then the phagosome, usually this all is an automatic process. So the phagosome, the pH in the phagosome drops, the phagosome fuses with other vesicles called lysosomes, and then you have the generation of a phagolysosome, that is this, and then we see that the bacterial cell no longer exists and is broken apart. And that is due to the dropping of the pH and due to enzymes that come in from the lysosomes that basically eat the bacterial cell alive. And then bits and pieces from the bacterial cell may be taken up by certain protein complexes. Here, the class 2 MHC protein. And then the loaded MHC protein is transported to the cell membrane and then presents whatever it has captured in these vesicles to the outside world. And here the rest is just thrown away. So for now, you could already start to remember that professional antigen-presenting cells they have the MHC class 2 proteins. Already as a spoiler, all cells, including those, have MHC class 1. We see what they do later. But the professional ones, only the professional ones, also have class 2. Okay, so here we have the cytokine-mediated immune response as well. And here is a rough depiction of how this works. So we have two cells here. So this is the nucleus of this cell, this is the nucleus of this cell. The nuclei are important here. What we have here is a virus. The virus infects this cell, gets into the cell, and multiplies. So we have lots of viruses. And then they are released by one way or another, and then they can infect other cells. And the process repeats, and then you have more and more viruses, and the host gets into trouble. So to stop this, this cell, through the pathogen-associated molecular patterns that come with the virus infection, can actually detect that something isn't quite right. And then this infected cell is able to activate genes that are normally not activated, and these genes include the genes for interference, for these messenger molecules. So this cell activates genes, makes interference, and releases the interference into the environment. So here we have the interference out. And alpha, beta, gamma just means that there are different types. And then the interference, they bind to particular interferon receptors and signal to cells that may not yet been affected that the virus is about to come. And then these cells in turn activate particular genes, and produce interferon-regulated proteins. And this is a little bit like if countries go from totally peaceful environment into a Cold War. So they don't want to have a standing army of hundreds of thousands of soldiers because it's very costly. So and it's the same with the interferon-regulated proteins. So we don't make them all the time. But if there is a threat that is imminent, then these wake up and make the interferon-regulated proteins. And if they are made before the virus infects these cells, then the virus will not be able to replicate or will have a much harder time to replicate. So that is how the interferon system works. Any questions? Good. Okay, and this is a footnote. The action is mostly carried by proteins, but there are certain microRNAs that can also be antiviral. So if we have a look what effect this has. So interferons are used or have been used to treat hepatitis viruses. And we're talking here about hepatitis B and hepatitis C viruses. And in both instances, there are millions of people that are infected worldwide. And interferons have been the drug of choice for Hep C, but as I told you, this has now been largely replaced by specific antivirals, but interferons are still the drug of choice for the treatment of hepatitis B. So, and this is something I showed you when we talked about hepatitis C, that if the patient is lucky, the interferon treatment can actually reduce the virus titer, reduce the damage in the liver, and even lead to the elimination of the virus from the patient, in this case, through the patient. Interferons can also be used to treat certain types of cancer, but not generally all cancers. But famously, the hairy cell leukemia is a type of cancer that is very sensitive to interferon treatments. However, interferon treatments are a little rough. So people experience a lot of side effects. So if you want to know how that feels, then remember the last time you had a flu, and not just a respiratory infection, like a full flu with clinical symptoms. And many of the things you experience with having like a real flu infection with disease, that is due to the fact that your body makes a lot of cytokines, including a lot of interferons. So that is how it feels if you are under interferon treatment. Okay, I also like to go a little bit into the signaling here. And I have this little dodgy animation. So what this is supposed to be the cross-section through a cell. And here we have the outside of the cell, here we have the cytoplasm, and here we have the nucleus. And in the nucleus, there are genes that are normally not expressed or not highly expressed, and they are expressed at higher levels in response to interferons. And these are the interferon receptors, and they are interferon receptors for different types of interferon, here, for example, for interferon alpha. And the binding of interferon alpha triggers a cascade that starts with the phosphorylation of kinases, so they cross-phosphorylate each other. So TYK2 and JAK1 are the ones in question. And they also phosphorylate sites on the receptor itself, which then can be bound by proteins that like sites that are phosphorylated. And these proteins are latent activators of transcription. So they're called, in this case, STAT1 and STAT2. So they bind the phosphorylated sites here, and then they get in reach of these kinases that basically phosphorylate, I wouldn't say everything, but a lot of things if it just comes close enough when they are phosphorylated themselves and active. And so they phosphorylate these latent transcription factors, which then can bind to each other, and then we have conformational changes that makes it possible for them to cross the nuclear membrane, to go through the pores in the nuclear membrane, and to enter the nucleus. And then they team up with other proteins, and then they are an active transcription factor, and then they bind to specific elements on certain genes. So they do not activate like everything. They activate certain promoters. And these promoters have to have an ISRE element, an interference sensitivity determining region. And if that happens, this gene is activated on fires. And that could be then a protein that is antivirally active. But it's a bit costly to make. There might be side effects. So therefore, we don't make it all the time. But we definitely should make it if there is interferon produced by virus-infected cells. Okay, and then a similar pathway exists for interferon gamma, but you can see here that it's the same scheme, but there are subtle differences. Here we have a homodimer of STAT1, whereas here we have a heterodimer of STAT1 and STAT2. So that means that this transcription factor here looks a little different, and therefore it activates different promoters. So again, this is the ability of the system to kind of direct the response in certain directions. It's not very specific, but it's somewhat specific. So, and then in response of all this, we have the interferon-regulated proteins that have these sensitive promoters. And these are the usual candidates. So I was for more than 10 years involved in the research of MX proteins. And they are very important because they allow us to survive influenza virus infections. All of us in this room, or I hope all of us, are functional MX proteins. Otherwise, you have real trouble in surviving the next best influenza virus infection. I actually hold at the moment a major grant to look into these proteins in sheep, because these proteins, some of them, are also responsible to fight retrovirus infections. In our case, HIV, and in case of sheep, other retroviruses, but sometimes they have veterinary importance, and people should know what they breed. So we found that many sheep in Australia and elsewhere have defects in these proteins, and the sheep would be more virus resistant if these defects would be eliminated. Okay, so a little bit on the history of MX proteins. So these, as I said, make carriers highly resistant to influenza. That was discovered by a guy called Jean Lindemann. So, and he published a paper on the resistance of mice to mouse-adapted influenza virus. And this guy here, Otto Haller, he got a paper in Cell, and I told you that Cell is a very important journal. And he showed that if you just take the gene for an MX protein and you transfer it to cells that don't have that one, then you confer resistance. So that even made it to the front cover of the journal Cell, and back in the day, that was basically your professor showed. So I got a little bit involved because this guy was my PhD supervisor, and I actually handled these mice, and you will see photos of mice that I took the photos. So one way to investigate these things is to make transgenic or congenic mice. And this, back in the day, was done by congenic mice. And congenic is a little different because you make two different mice that in the end are more or less the same except for a particular gene. And you can do this by conventional breeding. But it takes its sweet time. So these days, that's usually done in transgenic fashion, but back in the day, you could do it in congenic manner. So you have a donor and a recipient mouse, and the donor in our case was one with the functional MX gene, and the recipient was a mouse strain with a defective MX gene. So this one, highly susceptible to influenza, this one, highly resistant. But because the two strains have differences that are not just in the MX gene, there are differences all over. So it's very difficult to say this one is resistant against influenza because it has an intact MX gene. It also has intact lots of other things. So correlation is not causation. So what you do is you breed them, and then you get an F1 generation that's half this and half that. Then you look which one in this generation have the functional MX gene, and then you breed them again back on the negative strain. And you repeat this and repeat this and repeat this. And you largely, not completely, but largely, every single time you do this, you lose half of the genetic material from the donor strain. So what you end up with after 10 generations, F10, is a congenic mouse strain, and this is the way to write it, that is largely the genetic background of the susceptible mouse strain, with the exception of little bits and pieces of the donor, including the MX. So now, if this one has the same phenotype as this one, although it has a genetic background 99.99% of the other one, then you can be fairly certain that MX matters. And how much it matters, I show you here. This is a susceptible mouse, and this is a congenic resistance mouse with a functional gene from that strain. So you infect them with influenza-like virus, and these viruses are really killers. You do not have to wait very long. So after 72 hours, this mouse, it's what's called moribund. Body temperature has dropped to room temperature. The mouse is hurting and stopped eating, drinking, so these days you wouldn't be allowed to run the experiment that far. So this is basically a dying mouse, whereas the other mouse and all of his comrades did not even show signs of resistance. And what this set of images shows is that one gene, even if it's not an adaptive immune gene, it's an innate immune gene can decide whether animals live or die. So this is a pretty drastic outcome that's depending on just one gene. Okay, more recent research on MX proteins, for example, here published in Science, shows that there are also variations in our own MX genes that have dramatic consequences. So, for example, the people who turn out to be susceptible to avian influenza viruses, to pathogenic avian influenza viruses, they have likely, it's not always the case, but likely, they have a variation in their MX genes that is responsible for the susceptibility. So it's basically the same story as with the mice. Okay, now we come, and we have a few minutes left, to the adaptive immunity. So this is the one that is carried largely by T cells and B cells. And the T cells, they do the cell killing, and they also produce lots of cytokines. And the B cells, they're responsible for antibody production. So here we have another time course experiment. And we have the magnitude of the immune response, of the adaptive immune response, and the time. So here, it's basically a full month. And here we have things that happen, exposure to an antigen, to something foreign, and then a second exposure to the same antigen. And we have a primary response after the first exposure and a secondary response after the second exposure. And what I want to show you that the innate immune response, that is the one in light blue, is always the same. There's not much difference. So the innate immune response in that aspect is fairly dumb. It always does the same thing. It does it sometimes effectively, sometimes not so effectively. Sometimes you really need the innate immune response, otherwise you would be dead before you make an adaptive immune response, but the innate immune response doesn't learn. It's always the same. Whereas the adaptive immune response, you see, you get a delay after the first exposure, and then a certain response, and when the crisis is resolved, everything goes back to normal. But if you then are exposed to the same antigen a second time, the adaptive response is faster, no delay, and higher. And this, in a nutshell, is why we use vaccines. So we give people the first exposure without the risk, or largely without the risk. And then, if they then meet in the field the real pathogen later, they're prepared, and they make this much higher. So this is the basis for the concept of vaccination. And you really have to tell that to anyone who doesn't believe this. And this is not a matter, this is a matter of facts. So in my opinion, people aren't really entitled to have an opinion on this because it interferes with the common good. And it interferes dramatically with the common good. To basically kill people if you let their herd immunity drop. Okay, so now I try to give a bit of an overview about T cells and B cells. So here in green we have the B cells, and here in blue and in red we have the T cells, so B, T. The B cells, the developing B cells, they show the antibodies they will later produce as plasma cells already on the surface. So and they do this because they have to find an antigen. If they don't find an antigen, they will live for a certain amount of time and then they will die and no one will ever know of them. But if they find an antigen, and if they get conformational signal of certain, provided by certain T cells, then they are allowed to differentiate, to develop themselves, if you like, into these antibody-producing plasma cells. And then these antibody-producing plasma cells make lots of antibodies, and the same type that has previously been displayed on the surface of these early B cells. Okay, and then the antibodies bind bacteria, they bind viruses, and then that's how they do their job. The T cells, they are the cytotoxic T cells here depicted in pink or red. They go after virus-infected cells, and therefore kill the production sites of new viruses. And then we have the T helper cells. They can produce a lot of cytokines, which sometimes have antiviral activity, but most of the time they help to the cytotoxic T cells and the B cells to differentiate and then to produce large numbers, and in one case to produce even larger numbers of antibodies, and in the other case to produce these cells that kill virus-infected cells. Okay, so now this is an important slide because it introduces for the first time how you can discriminate between the cytotoxic T cells, TC, and the T helper cells, TH. You see, they look largely the same. If you look at them through the microscope, you will not be able to tell the difference. But they have different proteins on their surface. They both have the T cell receptor. That's why they're called T cells. But the core receptor is different. CD4 in the case of T helper cells, and CD8 in the case of cytotoxic T cells. This is important. You have to learn it. Okay. And here is an image that shows why it takes so long. And that is because we have a process that's called clonal selection. And this is shown here with T cells as an example. And I told you before that all immune cells originate in the bone marrow. And the T cells are actually called T cells because from the bone marrow they go to an organ that sits here, the thymus. And here they do something really strange, which we will discuss in a subsequent lecture. They are allowed to fiddle with the chromosome DNA, which normally is not allowed to happen. And that's for good reason, because if you allow cells to do this, you run a high risk of ending up with cancer cells. Nevertheless, these T cells can do it, and they do gene arrangements, and at the outcome of that, they have lots of different T cell receptors. So 10 to the 15th, that's a number that's very hard to comprehend. 10 to the 15th possible T cell receptors. You will not be able to make 10 to the 15 cells, not a fraction of this, but this is what you could make. This is a random process. And you have lots of these cells that have all different T cell receptors, but they all come in very small copy number. You may have just a single cell. And if that cell, by just the odd chance, then have a T cell receptor that binds to a virus, then this cell gets really excited and it gets support from other cells, the T helper cells, and then it is allowed to multiply. And then you make lots of cells of the one you basically randomly made with the fitting T cell receptor. So the equivalent is to defend yourself with a shotgun and you constantly shoot in all directions. You don't know what is coming, and then just hope that one of your little pellets hits. But when someone screams, then you just keep shooting into this direction. So this is more or less the metaphor for this. So it's also a completely wasteful process because all the other cells are not really good for anything. They're tossed away. So on the other hand, you prepare yourself for almost any possibility under the sun because 10 to the 15 is a pretty large number.
So the whole process can be described as completely random?
Elements of this are random.
Well, as it like...
Yeah, we will see if we come to the finer points. So, you know, if I say everything is random, then some people say, oh, this is not really completely random. So it is skewed in a way that you have variation and variability where it counts most. But there are certain elements of it that are completely random. Okay, here we have a slide, like basically enlargement of the whole antibody story. So we have the early B cell, we have the antigen. Here it's a bacterial cell or like a little bit on the surface of a bacterial cell. And then we have the differentiation into antibody-producing plasma cells, and then they do and go after the same bacteria that triggered it in the first place. B cells also mature in the bone marrow. A single B cell expresses only a single type of antibody, but can have lots of it, so 10 to the 5 molecules per cell, not just the 5 depicted here, but they're all the same. And random gene arrangements generates an enormous number of B cells with different antigenic specificities. You see it's a little lower than your T cell number, but still large, 10 to the 10. And then you produce the antibodies. And this is a clonal selection shown for B cells. You see the principle is very, very similar. So you do not have the thymus here, but you do have the gene arrangement. You have the huge variation in cells that come out of the gene arrangement. Then you have the lucky one that finds an antigen, here a viral antigen, and then this one is allowed to multiply and to differentiate. And here is another speciality. So you see you have the plasma cells that produce antibodies, you know this already, but you have one that's named memory cell. And this is why the adaptive immune system is a little smarter than the innate immune system, because you produce those and they are supposed to live for a long time. And if you are exposed to the same antigen again, then these ones start this process here much faster, much more efficiently. So that is the memory of the exposure to the antigen. Okay, largely in time. These are the learning objectives. And there is time for questions. And then there is a 5-minute toilet break. And then we go to the next lecture, which picks up some of the topics that I tried to introduce in this one. Okay, questions? I switch the lights on so everyone wakes up. That's all one.
So the clonal selection and expansion, that's in context of what you did with the mice here?
What? Say it again?
The clonal selection and expansion, is that in context of what you're doing with the mice?
No. Yeah, I mean, it happens in mice as well, but the experiment I showed you, or what we learned from the mice, that was in relation to an innate immune gene, the MX gene. And the clonal expansion is an element of the adaptive immune system. If no one runs to the loo, I could continue straight away. Is that a problem? Good? Okay, so now we'll talk about antigen presentation and recognition and all of this. So the subject is a little bit more narrow. So, and it hopefully fits into something I just told you in the previous lecture. So first, I would like to talk about antigen processing and presentation, and then we will talk about recognition. All right, so maybe we take our time with this one. Antigens are presented by MHC proteins. I said this before. Right, let's go to here. And here we have a virus-infected cell. And the virus-infected cell has viral antigens, so that's bits and pieces of the virus. So these are, for example, proteins that are not normally made by cells, but are made by these cells because the virus hijacks the protein making machinery. So, and these bits that aren't self, but they belong to the virus, are depicted here in red. And what you see here in green are these MHC class 1 molecules. So I told you before that we have two types of MHC molecules, class 1 and class 2. In this example, they are either green or orange. And I told you that the class 1 molecules, you can find them in all cells. Ordinary cells in the body that may or may not be virus infected, and the antigen-presenting cells, so here and here. Whereas the class 2 molecules, they're not made by ordinary cells. They're only made by the professional antigen-presenting cells, which makes the antigen-presenting cells having two MHC types, whereas the ordinary cells have just one. So, and this slide will show you what the different MHC classes do. So, if a virus infects a cell, the cell will automatically present bits and pieces of viral proteins on MHC proteins, and that's depicted here. It will always do it, and viruses have a very hard time preventing it. So therefore, the cell will actually show to the outside world that it has just been hijacked. And here we see two cytotoxic T cells that interact with these MHC proteins that show viral proteins or present viral proteins. And then these T cells will actually kill the virus-infected cell, and therefore the viruses within. In an antigen-presenting cell, they can be virus-infected, yes, but they also can eat viruses. So they can do phagocytosis. They can also eat bacteria. So they tend to eat all kinds of things they think do not belong to your body. And then they break it down, as I showed in the last lecture, and then they present this stuff on the MHC molecules. And these ones, the MHC class 2 molecules, they are not recognized by cytotoxic T cells because there's no need to kill these cells because they're not virus infected. They are recognized by T helper cells. And the T helper cells, they take the information and go away. They do not kill the presenter, whereas these cells kill the presenter. But then the T helper cells, through a mechanism become later, encourage certain cytotoxic T cells to multiply and to go out and kill virus-infected cells. Okay, important slide. Because it also shows which cells have class 1, class 2 MHCs, which cells have T cell receptors, and which cells have the T cell co-receptor, CD8 and CD4. So this is not random.
So just to clarify on the previous slide, with the antigen-presenting cells, the T helper cells come in contact with it. They don't kill it, of course, but then they go away and encourage the C cells to multiply, and that's also kill the virus-infected cells.
That's correct. They can do similar stuff with B cells. So they can depending on what T helper cell subpopulation you have, they then direct say, okay, we have a virus infection going on, we need more cytotoxic T cells. The cytotoxic T cells are pretty useless in a bacterial infection, so then hopefully you make an immune response that's more antibody driven. Cool. So now I have a couple of slides that show you the differences between MHC1 and MHC2. If you just squint your eyes a little bit and look at this, you think there's not that much difference, and that's true because they are related. But if you then look closer, you see there is some. So in green, MHC class 1, in red-orange, MHC class 2. So the green ones on the surface of nearly all cells, I showed that before, and they present endogenous antigens to CD8 positive cytotoxic T cells. And what endogenous antigens are, I'll show you in a minute. The MHC class 2 proteins, they're found on the surface of professional antigen-presenting cells, macrophages, dendritic cells, and also B cells. So these are the ones I produced, the APCs I introduced before. And they present exogenous antigens to CD4 positive T helper cells. And this is how they look like. So they both consist of 4 domains, but the distribution of the domains on individual protein chains is a little different. So you can see how the evolution has kind of worked on a protein that was an ancestor of these, and then you developed into having MHC1 and MHC2. Okay, so here we have the side view in this kind of depiction as I showed before, with a focus on how these things are attached to membranes. And here the side view in, you have beta sheets and alpha helixes, and here we have the top view. The top view is quite interesting because we see that we have a beta sheet here and 2 alpha helixes that confine a certain space. And this is where the antigen is bound. The antigen is bound here in the middle, here and here. And we already see that their space is a little different. Here's very rigid and small, and here's a bit more variable. So we come to this later. Here we have, again, MHC1, MHC2. I tried to keep with the color scheme here as well. And here we have antigens that are bound between the 2 alpha helices that constrain this space. You can, I don't know, that is provided by that beta sheet underneath. And you see that in case of MHC2, larger antigens can be bound because the whole thing is a little bigger. Okay, so that is depicted here. And so in the case of MHC1, we have certain anchor residues that really constrain this space. So all I'm telling you that these are not absolutely identical. Then this one explains where the name comes from. MHC stands for Major Histocompatibility Complex. And what you see here is, you do not really have to learn the details of this, is that MHC class 2 genes, you have lots of different copies. And this indicates that we have a huge variation amongst individuals. So my MHC molecules are most likely very different from the next best person in this room. So that means that as a population, we have lots and lots of different MHC proteins because we have lots and lots of different genetic elements from which we make the MHC proteins. And if some of us resist certain virus infections better than others, then we guarantee the survival. Whereas, on a population scale, we can always afford to lose a few. It's a bit unfortunate for individuals, but on a population scale, it's good if you have diversity. Okay, and this is something that's not my fault, but it makes it a little harder, and that is that the MHC is referred to as H2 complex in mice, and HLA in people. And if you go to the textbook or go to the internet or even go through my slides, so it's sometimes H2, sometimes HLA, and sometimes MHC. That largely means the same thing. This is just things that exist in parallel. So what I try to tell you is that the MHC genes are highly polymorphic. That means that more than one allele exists for a given gene. That means that we are most likely all different. And you see that, for example, class 1, the allotypes can give, a staggering number. So 2,480 proteins here, 3,221 there. So huge diversity. Don't learn the table, don't learn the absolute number, not necessary. But this is completely different from other genes you have. So there is also like an odd thing that this allelic variability is not just important for antigen binding, but also who you choose to procreate with. And this is one of the stranger bits of our textbook. And there's a chapter that's entitled Sweet Smell of Diversity. So people believe that you can sniff out the ideal partner. And that whether you like someone or not like someone, whether you're more likely to have sex and babies, depends on the MHC protein. And you will not, as a matter of how likely it is, you will not choose one that is exactly like you are. You will choose one that is as different as possible as you are, because that gives the offspring set of genes that are different and increases the chance of survival. So basically, we are geared to maintain the diversity and to have offspring that is heterozygous in these genes. And also, if you see where exactly these different alleles are different, then you see that here are the two alpha helixes and here are the beta sheet that define the space where antigens are bound. And you see the hotspots for diversity are spots that are very critical for binding different antigens. So the places that matter most have the highest degree of diversity. So it's not completely random, but it's kind of random. Okay, and now I like to explain the difference between endogenous and exogenous antigens and how they are processed and presented. So here we have the presentation of endogenous antigens via MHC1. So this is the MHC protein that exists in all kinds of cells. So here we have basically an ordinary body cell. It could be, I don't know, a cell in your muscle, in your liver, in your skin. And that has MHC1, but not MHC2. So then, and here we have a virus, and this particular virus, uses the nucleus for replication. But as you know, not all viruses do this. So in this particular example, it's just the case. So, and then in the end, the viruses rely on the cellular ribosomes to make their proteins, and then you end up with a viral protein that's here depicted in green. And that viral protein, that sometimes some copies of it end up in a process that I will explain in a minute, in the endoplasmatic reticulum. And they're picked up by these class 1 MHC molecules, and then these make their way to the cell surface through the Golgi and through vesicles, and here they are. And they are loaded with bits and pieces of that viral protein. So therefore the cell shows to everyone who's interested that it is virus infected, and that is then picked up eventually by cytotoxic T cells, and the cell has a fair chance of being killed. So here they have the presentation of exogenous antigens. So this is done by these professional immune cells only. So they also have the MHC1 proteins, but they don't play a role here, so they have been omitted. And here, these cells, as I showed you before, they can eat up things by endocytosis or phagocytosis, depending on how big things are. So if a bacterium is eaten by phagocytosis, for example, then it's broken down, as I explained before, and then these things eventually are loaded onto class 2 MHC proteins and presented. And then this signal is then picked up by T helper cells. So here we have the details of the process. Endogenous pathway to the left, exogenous pathway to the right. We start with the endogenous pathway. So we have a protein, likely from a virus, again depicted in green, and then you may think, why should this protein to go into the ER and then be presented? And that is happening because certain individual proteins end up being misfolded. They're basically rubbish. And then the rubbish is picked up by proteasomes, and here we have the depiction of a proteasome, and they chop the protein into little pieces, and these little pieces are then transported by TAP proteins into the ER, where they find their way on the class 1 molecules. So this illustrates that up to 30% of newly synthesized proteins are misfolded. Our own proteins and viral proteins. It just can't be helped. A certain number is just rubbish. And they are sometimes given a second chance to refold, and if it all fails, they need to be disposed of. And the job is that of the proteasome. And these misfolded proteins, they are made obvious because they get a tag, and the tagging is called ubiquitination. And this is actually the little tag. It's a 76 amino acid protein that is basically receiving this little tag. And sometimes it receives tag after tag after tag. So therefore, it becomes really, really obvious that this is a misfolded protein. And these tags are kind of the signal for the proteasomes to pick up these misfolded proteins. And the proteasomes are basically a little bit like a tube, and they have one end in which they feed the misfolded protein, and in the middle they have proteolytic activity. So protease just does clip, clip, clip. And so therefore they cut up the proteins. And that's how you get these little protein pieces. And the little protein pieces, they are transported via cellular, via specialized transporters, and they're called transporters associated with antigen presentation, or TAP. So here we have the protein, here we have the proteasome, here we have bits and pieces of the proteins that could either be further cleaved into amino acids and then used to make new proteins, or some of them are picked up by these TAP transporters and then forced into the ER. And then in the ER, they made the class 1 proteins, and that's shown here. That in itself is fairly complex. So here we have a MHC1 protein, and that means chaperones, so it actually doesn't go and picks up something it shouldn't pick up. And did you learn in biochemistry what the chaperone is? Tell me if you don't. Did you learn it? Does anyone know? Does anyone know the meaning of a chaperone? It's a very Victorian term.
Escorts, companies, guides, I'm not sure the exact word, but like a chaperone would take someone to the ball and make sure that they don't get into trouble.
Yeah, they don't get into trouble. That all the, the people with too much testosterone go after your innocent 15-year-old. So this is not going to happen. So therefore, you put an aunt in charge, and that is then in the Victorian times called a chaperone. Exactly that. And this is a function of these, Kinexine and ERP57. You do not have to learn this to the nth detail, but these are chaperones that bind to the MHC class 1 protein, so it doesn't, get into any trouble into the ER. So it doesn't bind anything it's not supposed to bind. And then eventually, the chaperones get exchanged and allow the class 1 protein to bind some of the peptides that are constantly transported into the ER. And then once they've lost the chaperones and they are loaded with a peptide, which is not necessarily a viral peptide, because this happens all the time, even if the cell isn't virus infected. So then they are allowed to exit and to eventually reach the cell surface and to present that peptide to the outside world. And then you may think, why aren't the cytotoxic T cells recognizing an MHC class 1 protein with a little peptide that comes from one of your own proteins? All innocent, no virus involved. And that is because the cytotoxic T cells that survive the clonal process, they are by definition recognize something foreign. They ignore all your own proteins. So this is going, again, a random process, again, really wasteful, because you constantly present stuff, constantly, all the time. But in almost all cases, it's your own stuff. And then nothing happens. But if a virus infects a cell, then in addition to your own stuff, the cell presents bits and pieces of viral proteins. And then the cytotoxic T cells recognize that and kill the cell. It's amazing how wasteful immunology is. Okay, so now you should know all what's happening here. And then we have, there's usually a pathway from the ER to the Golgi to the cell surface, and then we have presentation. And something similar happens here, but here it's usually not, because it's not endogenous. So it's not a protein that is produced by the very same cell. This is a protein that's eaten by the cell, so therefore it's the exogenous pathway. So we start again with an MHC protein in the endoplasmatic reticulum. You see the different number. You have an invariant chain here. You see this is some sort of chaperone. Goes to the Golgi, ends up in vesicles with that chaperone blocking it. But then in other types of vesicles, the chaperone gets degraded and the affinity for the MHC protein drops, and then the chaperone, in this case it's called CLIP, it's a class 2 associated invariant chain, is replaced by a peptide that comes, in this case here, from the bacterium that was eaten, and then ends up at the cell surface. Is that clear? Again, I can only do that much in explaining it, so this may require some reading in the textbook to just reinforce it. But the slides I'm using are largely from the textbook, so you should find it. Okay, so now I spent some time to explain how the antigens are processed, how they find their way on these two types of MHC proteins, and how they are then presented on the cell surface. And now we come to the recognition. So here, get your eye in. Here is a T helper cell. Here's an antigen-presenting cell. Here's a cytotoxic T cell membrane. And here's a target cell that's like an ordinary virus-infected cell. So here we have the antigens. In case of the antigen-presenting cell, it's an exogenous antigen, so they have been eaten, processed, and finds its way on the MHC class 2 protein. Here's antigen, here's MHC class 2. And I told you that this is specifically the target for the T helper cell because it makes interaction with the T cell receptor, that's this one here, and the specificity is provided by the CD4 protein, that's this one here, that really likes only class 2 proteins. So this is at the heart of it. There are other things going on which we now happily ignore. So here in this case, we have antigen presentation, for example, a viral protein misfolded, tagged, cleaved into little peptides, put into the ER, bound by class 1 MHC, and in keeping with all the color scheme the same of previous slides. So we have the antigen here, class 1 MHC here, that is also recognized by the T cell receptor. Specificity is provided by the CD8 protein. So and only the cytotoxic T cells have the CD8, and the T helper cells have the CD4. That is why they are so specific for class 2 MHC in this case, class 1 MHC in this case. Okay? Okay, now we come to different T populations again. So here we see something that you already know. You can relax. Here are the T helper cells. They have the T cell receptor and CD4. And here we have the cytotoxic T cells that have the T cell receptor and CD8. So, but then you will always find something in immunology and say, we found a population, in this case gamma delta T cells, that neither CD4 nor CD8 assists in the T cell receptor to bind something. So these have functions that are similar to cytotoxic T cells, but they do not need antigen presentation by MHC. So they are kind of evolutionary, more old school. They are more like natural killer cells. I'm just introducing this here, so you know, I'm already totally simplifying the story here. So if you go to the textbook and then you see, oh yeah, there are more T cells Michael never told us about, they even more. This is just one slide to show you that if you want to do immunology, you will have lots of these. Okay, this is me coming back to a slide we already know, and this is to illustrate that T helper cells play an important role because only B cells that receive additional activating signals, for example from T helper cells, develop further and survive and have a chance to become plasma cells / memory cells. So, and I showed you this before, and that shows, not by chance, the T helper cells here in a central position because they assist this development and this development. Okay, now we come to B cells. They have MHC1 and MHC2 proteins, which makes them professional antigen-presenting cells. But why? Because they need to present antigens to CTLs in the case of an infection, and they need to interact with T helper cells. So here, some important facts about B cells. There's a B cell, and I told you that the early B cells have the antibodies stuck on the cell surface. Okay, nothing new. I already told you they come from the bone marrow, and that B cells express only a single type of antibody. It's very, very important to remember, but they can have many copies. And this is largely the antibody they later going to produce if they have the chance to become plasma cells. Then I also told you that random gene arrangements generate an enormous number of different antigenic specificities. So it's a funny word of you can have 10 to the 10 different antibodies on B cells, but only one on one cell. So, but this is just the options you can have. And then there is something different again, and that's called somatic hypermutation. And that happens once you choose a new winner that will go clonal expansion. Then they are given a further chance to fiddle a little bit with their genetic material. What is this? Okay, well, complex subject, projector suffers. It's called cyberhypermutation, so that is further to increase antibody diversity by fiddling exactly with the antigen binding sites of these antibodies. Okay, so how does a T helper cell help a B cell? So this is supposed to be a bone, and here in the bone we have the differentiation from the stem cells, the gene arrangement into mature B cells that have their antibody already on the cell surface. So here in the bone marrow we have the gene arrangement. Then these cells leave the bone marrow and get into lymph and blood. And then they have to have further differentiation to eventually produce the plasma cells or the memory cells. And this happened in peripheral lymphoid organs, for example, in lymph nodes, but also in the spleen. So we have these naive B cells that are coming in here. They're randomly produced in the bone marrow. And you have like a huge diversity of those, but only a very low copy number, potentially just one cell. And if they don't find an antigen, nothing will bind them. These poor cells die. And that happens to 90% of them. If they do find an antigen, that's plus Ag here, which then consequently happen to only 10%, then they are relatively happy and then they will survive for a little longer. And if they continue to bind their antigen, and they will change a little bit, and then sometimes some can do it better than others, so there will be a little bit of elbow and competition, then they eventually can become plasma cells and memory B cells. Okay, so and to be amongst the lucky ones here, there is the word T helper cell. So they need, in addition to binding the antigen, they also need to please a B cell. And this B cell can only be pleased if the antigen they are finding is taken up. So here the antibody was by chance binding the antigen, and then this antigen is phagocytosed, that makes them professional antigen presenting cell, processed, and then ends up on MHC class II proteins. And that can be recognized by the T helper cells, and so that makes this particular T helper cell happy, so activates the T helper cell, and then the T helper cell produces proteins that can strengthen the interaction, and it produces cytokines that can be detected by this B cell. So all in all, the B cell needs 3 signals. One is provided by finding its antigen. The second one is provided by making further contact with the T helper cell through receptor-ligand interaction. And the third one is provided by detecting cytokines produced by that T cell. So 1, 2, 3. And if they have 1, 2, 3, then this one will divide and divide and divide and makes many, many copies of itself. So it's basically a double check whether a naive B cell is the chosen one and can undergo clonal expansion. Depends on whether it finds its antigen and whether it gets conformational signal from a T helper cell. Only then will this cell survive and eventually produce antibodies. So you see, there are a lot of checks, and that is because if you make an antibody against your own protein, you are seriously sick or dead. So this should not happen. And that's why you have all these checks and balances in the system, which makes it very hard to learn. Yep.
Could you really quickly explain just it's the second signal, the CD40, how does that work again?
Okay, I think I have subsequently a slide on just this. So basically there is a bit of a staggered response. So the first signals kind of increase the process, the expression of some of these, and only then they happen. But you will get a very good mark if you already know what's just in this slide. Okay, clonal selection of B cells. So this all fits into this. The whole idea is to have so many checks and balances that this is happening only if there is indeed a foreign antigen that potentially is dangerous. So, and then T cells drive direct type of immune responses, for example, towards an antiviral response. So here we have our activated B cell, and here we have it undergoing the clonal extension, and here we have a couple of plasma cells, and they all produce antibodies, but they produce slightly different antibodies. So we will have a lecture more on antibodies, but I can tell you already that we have different classes and different subclasses. So IgM and IgG, There are two different classes, and IgG2 and IgG3 are different subclasses. So what you need to learn here is that they produce antibodies, but the antibodies are a little different. And some antibodies are more useful to fight virus infections, whereas other antibodies are more useful to fight parasite infections. So, and to get the right outcome, you activate and please different T helper cell populations. So for example, T helper 1 and T helper 2. And then these decide whether these cells become plasma cells that, for example, make IgE, parasite infection, or IgG2A, typical for a virus infection. And this is done by different cytokines. So it's not just interferon with that crude antiviral response I showed you in the last lecture. These are interleukin-4, for example, for T helper 2, and interferon gamma for T helper 1 response. These are the dominating cytokines that drive the antibody response towards a certain direction. Okay, we leave it as, we leave it with this slide as that. So what you may want to learn is that the T helper 1 response is typical for virus infections, and the T helper 2 is typical for immune responses against parasites. This has some consequences in a way that early on in the history of mankind, we had to find many more parasites than we do now, because we have a bathroom and we have a toilet, and that's where the poo goes. And we have a kitchen and a sink and tap water, and that's what we drink. This wasn't previously so much separated. People would drink from the river, and they would also kind of defecate next to the river. So, and this would all meddle a little bit up, and that was very congestive to parasite infections. So the T helper 2 response got a little out of fashion. So, and then that is the consequence of people, or it's believed to be the consequence of people having more autoimmune problems these days than they had perhaps 5,000 years ago. So to study these things is not just academic, so this is really important. Okay, and this is all I wanted to say here for the learning objectives. Yes, that's it. But I am absolutely happy to take questions, and you should have questions. Yes?
Antigen-presenting cells have both the MHC proteins. What stops a T cell from killing the presenting cell?
Okay, good question. So the same reason that stops the T cell of randomly killing cells in your body. The T cells, okay, you have the T cells that do the shuffling of the genetic elements in the thymus and have the random generation of this vast amount of possible T cells that leave the thymus. However, in the thymus, you have the T cells that are being generated at random, and of course it's random. Some of the T cell receptor will bind your own antigens. But if that happens in the thymus, and in the thymus there are other cells that kind of try to expose the T cells to as many of their own antigens as ever possible. If the T cells do this in the thymus, they get killed. So almost all T cells that go that genetic shuffling die in the thymus. They're generated, but then they die quickly afterwards. And the ones that are allowed to leave the thymus have been selected so that their T cell receptor doesn't actually recognize anything. So, and then they wander around, and most of those will in their whole life not see anything and get excited and die. And only if there is a virus in the body, which does not always happen, and if the virus just by chance matches one of the T cell receptors of the T cells that survive the vigorous killing of T cells in the thymus, then the clonal expansion happens. Does it answer your question? Very good question. Do we have more of them? Yes.
Do the tap proteins that move the peptides,
Yes.
VR, is that for a different reason, or do they just do that from a regular process?
Yeah, so the whole thing is the body does not know what virus comes next, and it certainly does not know what virus will come in 500 years or 4,000 years. So the whole process is basically has these random elements. So basically, misfolded proteins get chopped up, as a standard procedure, and then some of the chopped up pieces, as a standard procedure, go to the ER. They are loaded and presented, and so it's very hard for the virus to avoid it. So there is, again, it's not always completely random. So for example, if cells are exposed to cytokines that signal an infection, then they make slightly different proteasomes. They're called immune proteasomes. And then the whole process is a bit more efficient, because then they kind of maybe chop up more proteins, things like this. So yes, it's slightly tweaked, but they do it all the time.
Do I understand correctly that 30% of rubbish proteins that are created, that they're essentially just chopped up and recycled and reused?
Yeah, don't get up to 30%. So that depends a little bit on the protein. Some fold more easily, others fold not so easily. Some are bigger, some are smaller. So I think I do not know the absolute number for every group. I doubt this is pretty much known, but fair to say and good to remember that a certain percentage of newly folded proteins have trouble folding correctly, and then they need to be disposed of. Yeah.
And in the process of disposal, they're pretty much recycled,
Yeah, it's everything that doesn't end up on a MHC protein, and only very few bits and pieces end up on MHC protein. I can imagine that the majority just gets recycled. So it gets broken down to the level of amino acids, and then you can make different proteins out of those again. I'm still happily here answering questions if you want to. So we have, because we basically made a back-to-back, we have plenty of time. I can even go back to slides that gave you trouble. IgE? Yeah. Yes.
Isn't type 2 have a sensitivity allergy response? Or?
Yes, but it wasn't designed to give you an allergy. So this one, this one was designed to fight parasites, but then if there aren't any parasites, then this part of the immune system is a little like a loose cannon, so it has nothing to do, and maybe it gets stuck into something it shouldn't get stuck, and then you get an autoimmune disease. So there is also the idea that, which is very complex, and I do not really want to talk against vaccines, but so when children get exposed to viruses and bacteria and all of this, that kind of trains the immune system a little bit and provides a certain balance between Th1 and Th2 T helper cell responses. So you want your children to face certain immune challenges. So that's the idea. So it's a kind of a hygiene theory. So yeah, you can do this by letting them experience polio and chickenpox and all this, and in the process of training them up, you will just lose a few. So that was the original idea, but this is not what we consider in a competent society what we should continue to do. So you do not really want to have people running around with feet that are damaged by polio, and people with having varicella zoster virus infections later, or people with running around with SSP and taking 6 months to die of a measles virus infection. So you don't want this. However, you still want them to develop proper T helper 1, T helper 2 balances. So it seems that people who grow up in the countryside, people who grow up with several children, and people who grew up, with the children in the house and having lots of neighbors coming over, and they tend to have fewer autoimmune problems. So because they're exposed to all kinds of things, and if you spray Dettol on every surface as it is suggested on all these commercials, and just provide a clean as possible environment for the children, you may not always do the best in the long run. So, however, I'm not saying to, let your kids eat the poo of your dog, so just to give them a challenge every now and then. So this is the other extreme. So there has to be a balance for this. And so therefore, in the long run, in my opinion, it might be good to have vaccines that produce a few side effects and give you a little bit of a fever, and perhaps even give you a little headache and some pain, because you want the challenge, but not the kill. And so therefore, in my opinion, it's not like absolutely desirable to have vaccines that do their job without also educating the T helper 1, T helper 2 population, so you find a balanced response for later stages of life. But this is a subject matter that's a bit in flux. Yes, but this is something probably as a young parent you may want to consider. Okay, some people are grinning. I hope I'm on, preempted something here. Okay, so any other questions? If this is not the case, then we close today and let's have a look. We have, the next ones are a lecture on T cells, and I will try to enforce some of the basics I already told you today, and then we have a lecture on B cells, and you will hear in all its glory how this all happens with the differentiation and the killing in the thymus, or the B cells experience the same wave of killing in the bone marrow, because these are basically like sisters or brothers, so they are not that different. And I will also tell you a lot about antibodies in one of these lectures on the B cell studies. And then we have one on cancer. And then you guys decided to hear in the very last lecture on my research projects, so that I do not sit here with 1 or 2 students. Even if I tell you about fossils, there will be a slide where I go back on bacteria and the difference between gram-positive and gram-negative bacteria. And this may or may not be the subject of a multiple choice question. So just because the topic is slightly left field, it's not completely irrelevant. So just to ensure that we have a little bit of this. And also, next week, Friday, and I will post it and make an announcement, we will have a guest speaker in medical science, Katharina Richter, as you can tell, a German-born scientist. And she has alternative ways to fight bacterial infections or bacterial contaminations. And she will receive a science prize here in Canberra and gives a lecture here at UC. And yeah, there is a lecture, and it seems she's a good speaker. Please come and enjoy a lecture that is for free, and the faculty will provide some food that's also for free. So this is all happening, I think, next Friday, but I will upload a flyer on Canvas. Yes?
Is that the lecture in place of these classes lectures or afterwards?
No, this is completely voluntary. So whatever Katharina says, and I don't even know whether she says anything sensible or not, because I never listened to her before, but my best guess is, and especially because she got a prize, that it must be something sensible. And she seems to be from what I gather online, seems to be a good presenter. But we will all figure this out on Friday. But this is just for your amusement or further education, but has nothing to do with any assessment that's coming up. You're a little late to the game. We're closing shop here. Okay, that's it. Have a nice weekend, and remember, no pracs today. So we follow this thing here. Huh? What do we have? No, if you look in this, it says no practicals.